Welcome back. We have learned that amino acids are linked by peptide bonds to form a polypeptide chain or a protein. Each protein has a distinctive number and sequence of amino acid residues, which determines how it falls up into its unique three-dimensional structure, and this in turn determines the function of the protein. The complicated structure of proteins can be conceptually described at four levels, including primary structure, secondary structure, tertiary, and quaternary structure. The last three are the three-dimensional structure of proteins. In this and the following sections, we will introduce the characteristics of different levels of protein structures. Here in this section, let's first discuss the primary structure of proteins. The primary structure of a protein refers to the sequence of amino acids from N-terminus to C-terminus in a polypeptide chain linked by peptide bonds, including the description of the disulfide bonds. Some observations illustrate that the function of a protein depends on its amino acid sequence. First, as we have already noted, proteins with different functions always have different amino acid sequences. Second, on comparing functionally similar proteins from different species, people find that these proteins often have similar amino acid sequences. For example, on comparing the insulins from different species, the A chain of insulin is identical in human, pig, dog, rabbit, and spur whale insulins. The B chains of the cow, pig, dog, goat, and horse are identical. They all have similar physiological functions in regulating metabolism. In other words, insulin from pigs can also work in human bodies. Before the early 1980s, almost all insulin used in diabetes treatment was extracted from the pancreas of pigs. By the early 1980s, people had successfully produced human insulin in large quantities from microorganisms by using genetic engineering technology. Third, if two organisms are closely related, the sequences of their genes and proteins should be similar. The sequences increasingly diverge as the evolutionary distance between two organisms increases. Therefore, amino acid sequence in the primary structure of proteins can provide important evolutionary information. By comparing the primary structure of proteins that widely distribute among different species, we can understand the evolutionary relationship between species. For example, the beta chain of hemoglobin contains 146 amino acid residues. Compared with humans, the differences of amino acid sequence in gorilla is only one amino acid residue. The differences between humans and frogs is up to 67 amino acids. Therefore, the evolutionary distance between frogs and human species is far away. Finally, thousands of human genetic diseases have close relationships with defective proteins. The defect can range from a single change in the amino acid sequence to deletion of a large portion of the polypeptide chain. Although the amino acid sequence in some regions of the primary structure might vary considerably without affecting biological function, most proteins contain crucial regions that are essential to their function. Gene mutations in these crucial regions can lead to the changes of key amino acids, therefore affect the structure of proteins and result in molecular diseases. Let's take an example to show how the variation of amino acid sequence can lead to disease. Please compare these two pictures. What's the difference? The left picture shows the normal red blood cells in human body with a uniform cup shape. On the right picture, the red blood cells are deformed and present variable shape, which range from normal to spiny or sickle-shaped. These cells occur in the blood of patients with sickle cell anemia, a hereditary human disease. The mechanism of this disease demonstrated strikingly the importance of amino acid sequence in determining the conformation structures of proteins and thus their biological functions. So, what happened to these patients? The red blood cells in patients with sickle cell anemia contain many abnormal hemoglobins, called hemoglobin S or HbS. Hemoglobin is a tetramer protein that is responsible for oxygen transport in the bloodstream. It contains 4 polypeptide chains, called globin, and 4 hem prosthetic groups. The globin consists of 2 alpha chains and 2 beta chains. Normal hemoglobin, called hemoglobin A or HbA, are soluble in red blood cells. When hemoglobin S is deoxygenated, it becomes insoluble and forms polymers that aggregate into tubular fibers. Hemoglobin A remains soluble on deoxygenation. The insoluble fibers of deoxygenated HbS cause the deformed sickle shape of the red blood cells, and the proportion of sickled cells increases greatly as blood is deoxygenated. The altered properties of HbS result from a single amino acid substitution. Normal hemoglobin has a glutamate residue at position 6 in the 2 beta chains, but hemoglobin S has a valine instead of glutamate residue. The R group of valine has no electric charge, whereas glutamate has a negative charge at pH 7.4. Hemoglobin S therefore has 2 fewer negative charges than HbA. Replacement of the glutamate residue by valine creates a sticky hydrophobic contact point at position 6 of the beta chain, which is on the outer surface of the molecule. These sticky spots cause the deoxy-HbS molecules to associate abnormally with each other, forming the long, fibrous aggregates characteristic of this disorder. The sickle cells are very fragile and rupture easily. This results in anemia. An even more serious consequence is that capillaries becomes blocked by the long abnormally shaped cells, causing severe pain and interfering with normal organ function. Sickle cell anemia is life-threatening and painful. People with this disease suffer repeated crises resulting from physical exertion. They become weak, dizzy, and short of breath, and they also experience heart murmurs and an increased pulse rate. So in summary, differences in amino acid composition and sequence of protein result in differences in protein function. Protein sequences are a rich source of information about protein structure and function, as well as the evolution of life on Earth. Sickle cell anemia is a genetic disease caused by a single amino acid substitution in each beta chain of hemoglobin. The change produces a hydrophobic patch on the surface of the hemoglobin that causes the molecules to aggregate into bundles of fibers and result in serious medical complications.